you for the presentation, and thank you all for being here. And today I'm going to present our work entitled Integrating Taxonomic and Functional Features for Gut Microbiome Health Indexing. So we have already assessed the importance of gut microbiome. We know it is important for metabolism regulation, immune response, and even the brain function through the gut-brain axis. And we know that thanks to the advances in metagenomic sequencing, we have now non-invasive and affordable methods to profile the microbiome. So it is important to build these health indexes. And the problem is that most current indexes are based only on the taxonomic profile, as it was already mentioned. So in this context, the CAMDA 2025 challenge aims to create a new generation of health indexes based not only on taxonomy, but also on the functional features. So in order to do this, we received a data set of 4,398 samples with their metadata. They were provided by CAMDA, and they were retrieved from the metagenomics data database. From this data, we obtained the pre-computed taxonomic profile, the pre-computed functional profile, and some metadata that contain the sample ID, the category where the condition is, and the cohort from the sample where it was taken off. Okay, so before starting the analysis, we performed an early review of the dataset, and we identified that there were some inconsistencies between the data provided from CAMDA and the original metagenomics data metadata. So, for example, we have some samples on the right, which were inconsistent from the metadata of CAMDA and the metadata of the original database. So what we did first was a manual curation of the data, where we went and checked the original publication of the cohort so we could correct those discrepancies. At the end, we could correct some of them, but still there were some uncertain cases that at the end were left out of the project. So we ended up with a final data set of 4,225 samples that were correctly labeled. So after that, what we did is that due to the high diversity of diseases in the samples, we decided to create 6 disease categories based on the International Classification of Disease framework. So the created categories were the following. First, we have the healthy group, then the metabolic disorders groups, neoplasms, cardiovascular diseases, infectious diseases, and mental or behavioral disorders. And so here's the distribution of the samples across all the categories created. And we can see that we have an unbalancement across all the categories compared to the healthy group. So after that, we perform an exploratory analysis by calculating the alpha and beta diversities, where we really didn't see any significant differences. Although the infectious disease group show a slight divergence, as you can see on both the Shannon index and the beta diversity plot. And now moving on to the functional profile, we did something similar. We grouped the metabolic pathways into 12 categories to focus on broader categories and improve the interpretability further on the project. And so there's also an example of one of the groups, which is the macromolecule modification pathways. And we can see how there are some differences across the groups of this pathway group. And so how do we integrate these 12 pathway groups with the taxonomic indexes? So the proposed workflow that we performed was the following. So we take the healthy group and a certain disease group. So as you already saw, it is unbalanced. So first we perform a data balancing process using an isolation forest to not select just a random subset of the healthy samples, but to select a really representative subsample of the healthy group. So after having both classes balanced, we retrained the original metagenomic indexes, health indexes, which are the gut microbiome health index and the HIPCA. And now to integrate those indexes with the 12 pathway group abundances that we have just calculated, we fit them all into a logistic regression model with an L2 regularization. And finally, we assess the performance of the model using a 5-fold cross-validation and the F1 score. So these are the results of the model. And as we can see, there are certain groups in which this ensemble model outperforms the classic taxonomic indexes. But it is also important to note that, for example, we have a group like the neoplasms, which actually performs worse than the taxonomic-only indexes. Okay, but focusing on the best performing groups, which it was, for example, the infectious disease group, we also wanted to assess which were the most informative features. So we looked at the regression coefficients of the logistic regression, and these were the most informative features. In the first place, it was the gut microbiome health index, but it was surprising that in the second place, it was one of the groups of the pathways that was the nucleotide and nucleic acid metabolism. And it was also interesting that we found some evidence that this is indeed related to this kind of diseases. We found that, for example, in diarrheal diseases, there's a stimulation in the production of cyclic adenosine monophosphate, and that triggers inflammation, weakens gut barrier integrity. So it is also great to know that this is not only statistically important, but it's also biologically significant. And so we do the same with the second best performing model, which was the mental and or behavioral disorders. And in this case, we also got as the first place, as the most informative feature, the gut microbiome health index. And then we got also some of the pathway groups that we created. First, the biosynthesis of cofactors and vitamins, which indeed we also found evidence that is related to this kind of diseases. For example, in the biosynthesis of vitamin B, which is linked to which its dysbiosis is linked to neurodegeneration and some mental health conditions like the Parkinson disease. Also, in the metabolism of amino acids, we found evidence. For example, we know that it is important that the metabolism of amino acids like tryptophan play a key role in modulating some neurotransmitter synthesis like dopamine. So it is linked to some diseases like depression and anxiety. Okay, and now moving on to another part of the work, we also wanted to analyze the interactions between the taxonomy and the functions. So what we did is that we built group-specific co-occurrence networks from the taxonomic and the functional profiles. So in order to do this, we tried different methods depending on the nature of the data. For example, in the taxonomic profile, as we had compositional type of data, we used the SpeakEasy library that uses a center block radio transformation and then calculates the inverse of covariance. And with that, we could build the co-occurrence network. On the functional profile side, as we didn't have that compositional type of nature, we just used the Spearman correlation and we built just the regular co-occurrence network. And so here are the results of the co-occurrence networks. Here's a snapshot of the most highly connected species on the network. In parentheses, you can see the degree of each species. And what's interesting about this is that every set of of group disease has its own unique set of species that were identified as hub species or highly connected species. So this means that actually these hub species characterize each group of disease, and this is pretty interesting. And also what's interesting is that some of these species that are marked with an asterisk are, well, we found evidence that they are related to this kind of diseases. And there is the species for the rest of the disease groups, the metabolic disorders, neoplasm, and cardiovascular. And so after that, we wanted to analyze the functional hubs, the hubs from the pathway group co-occurrence networks. So this was a surprise because it presented another type of behavior. For example, we could see that unlike the taxonomy hubs, we found some functional hubs that were shared among many groups. But we also find that there were some hubs that they were clearly differential in certain groups. And we also found this glycolysis glyoxylate bypass pathway, which is a strange case, because it is a functional hub throughout all the disease groups. So taking this in mind, we think that for the future work, we would like to incorporate this co-occurrence information to the model. And how are we planning to do that? Well, we would like to add, for example, the presence or absence of hubs as a feature for the model. And maybe we are also thinking about detecting communities, which are a set of really interconnected species in the network or pathways. And we can also add that as a feature for the model and see if it improves the prediction. Also, another direction that we want to take is extending the existing indexes for multi-class prediction. As we already saw, these indexes are meant for binary classification, but we are thinking that maybe using a strategy called 1 versus 1, we could make this or turn this into a multi-class classification problem. And we have some preliminary results that are very encouraging. So we think that maybe, although the problem seems more difficult, maybe it will have better results. And also for the future work, we would like to consider population origin. We made just like an extra analysis where we added some Mexican samples to the CAMDA samples and calculated the beta diversity. These Mexican samples are from the Latin Biota Consortium. And what we see is that there's a clear difference between the CAMDA samples and the Mexican samples. This not only highlights the underrepresentation of Mexican and Latin American samples in cohort data, but also tells us that the geographic origin of the sample is a big source of variation. So it needs to be taken in consideration for the next models. Okay, and finally, I'm excited to introduce our contribution to the microbiome research community, which is our web tool called the Gut Microbiome Health Calculator, which is a tool for the easy calculation and continuous evaluation of these kind of models. As right now, it hosts the best models presented, as well as the original models and the model from the last year's data. And we invite all of you to use it and test it on your own data, and please give us feedback. So in conclusion, what did we do here? We developed an ensemble model that successfully integrates both functional and taxonomic features, at least for two of the presented groups, which were the infectious disease group and the mental or behavioral disease group. Also, we built co-occurrence network that revealed several hub species that were differential among groups and that could be integrated further in the indexes. And also, we did the same for the functional pathways, but we found that the functional pathways are kind of different, so they are shared among the disease groups. Okay, and finally, I just want to thank all the people that were involved in the development of this work. This was a collaboration between two Mexican institutions, the Universidad Nacional Autónoma de México and the ISMB staff. There's also the QR code for the GitHub repository if you want to take a look at the scripts and the results. And I also want to thank the ISCB for giving me this opportunity to present my work. Thank you.
Thank you very much. I think it's really exciting that you also managed to create a publicly available tool. Have we got any questions? Okay, maybe I will start. I'm particularly interested about the classification of the CAMDA versus Mexican cohorts. Did you calculate this only on taxonomy, or did you use functions too?
No, it was only on taxonomy.
Yeah, I think this is the premise that we've been trying to push. I wonder if you calculated this on functions, whether you would see more overlap of the two.
Yeah, sure. I should do it, although, yeah, but I haven't.
Yeah, I think it would be really interesting. Okay, any more questions? Yes, there is one.
Hello, nice talk, thank you. I have a question relating to the co-occurrence network between the function and the taxonomic profile. So you use Metaflan and Newman, right, to get the two information. But did you try at some point to figure out from a presence absence of those genes if the function were really into those, or it's just completely agnostic and you take you don't associate the function to a specific bacteria? Or is it just from the co-occurrence network that you look at that?
Can you repeat the last part, please?
So I'm wondering if your taxonomic profile and if the functional profile are linked only by the co-occurrence or also by genomic evidence.
No, they're just linked by the co-occurrence.
Okay. And do you try to look at those kind of information, like looking at the genomic information from your taxonomic profile and see if the function are inside the reference of those genomes, for example?
No, I think I haven't, but I think it's a good idea. I think we should do it, but right now I didn't do it.
Okay, thank you.
There's one online question. So online they're asking if you think that the differences between the CAMDA cohort and the Mexican cohort is due to different library protocols, like sequencing, different sequencing applied.
I mean, it could be, but well, as the CAMDA cohort, it was pre-computed. I don't know exactly like which were the sequencing parameters they used. I know about the Mexican data, but maybe there's maybe there's a variation because of that. But still, I think that even though they were sequenced the same, still there will be some variation.
Great, thank you. If there are no more questions, then I think we can all go for a break, and we will see you back shortly.